Yeah, good afternoon, everybody. And it is really a privilege and pleasure to be here this afternoon and share a little bit of the journey that I had from doing a PhD in biophysics to eventually becoming an entrepreneur and developing new red blood cell-based therapies. Now, I want to start with a little bit of my background story because you may hear it on my accent, but I'm neither from Canada nor from Australia. I'm actually from a small town in southwest Germany, right at the French-German border. And this area is known for a beautiful landscape, excellent food, but most importantly, it had the biophysics program that I enrolled into, and we were only like 20 students. And this kind of close environment made it ideal to kind of learn with close interactions from the professors, all the details of biophysics. But when you live in such a small, tiny village, you actually want to explore the world a little bit further relatively quickly. So I decided to move across the Atlantic and ended up in Toronto, where I eventually completed my PhD in 2021. And this PhD was focusing on particle physics. Especially, I used particle physics as a tool of microscopy in order to study the molecular and structures of red blood cell membranes. Now, you may all be familiar with traditional light microscopes, where you can see here on the left-hand side the microscope picture of cells. But with a light microscope, you cannot look smaller and smaller. In order to see tinier structures, you actually need to use particles in order to resolve proteins and membranes and other molecular pictures. In my PhD, I used particle accelerators in order to study the structure of red blood cell membranes. And the idea is pretty simple. You take a particle accelerator or a nuclear reactor, you shoot the particles at the material you want to study, and what we as a physicist do, we actually look at how these particles disperse from the material in order to understand how they look on the molecular level. And for the first time, I was able to measure a molecular high-resolution image of a red blood cell membrane. And this gave me insight into all the details of how these membranes look like, how they move, how they organize themselves. And this was, at the time, the most detailed image of a red blood cell membrane that we had, and which became eventually critical to the work of what we're doing at Synth-Med Biotechnology now. Doing research on blood requires, first of all, a lot of blood. And then, well, if you're wondering where all this blood is coming from, well, I was quite literally bleeding for my PhD. And if you're wondering how this sounds like, I don't know if the audio was going. Anyways, they missed the veins quite often, and I had a number of blood. So, aside from bleeding for my PhD, I'm also quite passionate about physics. I'm passionate about programming, biochemistry, tinkering, and creativity. And all of these kind of skills, interests, and multiple disciplines is what brings us together at Synth-Med Biotechnology, where we actually innovate the world of drug delivery. Now, if you take a pill, say, Tylenol or Advil, for instance, the drug actually distributes itself all across your body. Now, for ibuprofen, that's not a problem, but for many drugs, you actually want the drug to go to the target of where it is needed. You want to have it ideally in your brain, in your lung, depending on where the cure is needed. And for many drugs that are currently in the clinics, a lot of them actually fail because they have a short half-life in the circulation, they have a high off-target activity, or they have a high dosing requirement. And this eventually lets a lot of drugs fail in the last phases of the clinical trials and makes something that is actually potent unusable. So in an ideal world, we want to bring this drug just to where they where they are needed, such that they can fulfill the therapeutic purpose, say in your brain, but leave the rest of your body unattached. And now our vision is to use red blood cells to bring the drugs to unreachable corners of your human body. Red blood cells are the natural transport mechanism that all of us have. They transport oxygen, nutrients, and they are highly biocompatible, non-immunogenic, and have a long circulation half-time. So ideally, so pretty much the ideal transport mechanism that your body creates. But unfortunately, the red blood cells on their own are not specific enough to bring any drug to the target that you want to deliver it to. So we need to outsmart the system in order to get the red blood cells being useful and being functional. And now comes into place what I did on my PhD. We studied the membrane structure on a molecular level, which means we can actually also tune and manipulate the membrane of these red blood cells on really the tiniest levels possible. What we develop at Synth-Med are nanocarriers, about 1,000 times smaller than a human hair. They use the membrane of red blood cells, but we dope the membrane with additional synthetic compounds that eventually enable us to load a therapeutic compound inside these nanocarriers and put a targeting moiety on the surface. And both of this allows us to eventually bring a therapeutic compound to the target that we want to deliver it to. We were talking about how long such an innovation actually takes, and I can tell you that I started my PhD more than 10 years ago, and this is about the time that it took in order to get to where we are today. We had some remarkable successes along the way. We developed a COVID vaccine during the pandemic that was based on red blood cells and delivered antibiotics successfully to bacteria. But at Synth-Med, we are focusing nowadays on two major challenges: on delivering drugs to the brain and to improve the outcome for cancer patients. The brain is one of your most precious organs, as such it is highly protected by a barrier called the blood-brain barrier. You get oxygen through it, you get carbon dioxide through it, and also the good stuff like alcohol and caffeine, but fortunately it protects pathogens from reaching the brain. And then on the unfortunate side, it also protects many drugs from reaching the brain. And overcoming this challenge is one of the most difficult ones in modern drug development. Now, we have developed carriers using our technology that actually can penetrate this blood-brain barrier. By using our red blood cell-based therapeutics, they circulate in the body, you just do an IV injection, and eventually bring the drug into the brain. The drug that we are loading into our carriers is a memory-active compound. Now, it is an endogenous protein that is present in all of your brains right now. And as your memory forms, when you're listening to my presentation, it actually works on forming the synapses in your brain. Now, if you have Alzheimer's disease, the concentration of this compound is lowered in your brain. And the idea that we initially had was very simple. We just bring additional amount of this protein back into your brain and hopefully can offset this memory loss. We are fortunate of working together with one of the leading scientists in the world on this particular protein that we're using. And with her advice, we were able to run preclinical tests on animal models. I'm just showing you how we actually test memory in animals. A mouse is not the smartest animal around, but we can test their memory by putting them in a pool. See here, like a water pool, and the mouse is placed into the water and then finds this island that is here, like circled in the red dot. The mouse itself doesn't see the island, but it sees this black dot that we put on it. And the mouse relatively quickly identifies this is a safe spot to go and eventually finds the island within a couple of seconds. In this process, the mouse actually memorizes how to get to this island. So then one day later, we remove this marker from the island. The mouse can no longer see where to go and how to get to this island. But it needs to rely on its memory to find this island and the safe spot in the pool. Now, if you have, I think some of the videos got mixed up actually in the slides. So what you see here is a mouse that has Alzheimer's disease. And this mouse actually takes about 120 minutes in order to circle in the pool, and it never finds the island. in this time. If you run the same experiments with a healthy mouse in comparison, you can see within just a few seconds, this mouse remembers finding this island without any difficulty. Now let's look again at a mouse that has Alzheimer's disease, but has been treated with our technology. Again, reminder, we bring this memory controlling compound back into the brain of the mouse. And within just a few seconds, this mouse has no problem finding this island again. And we have tested this with more, thank you. We have tested this with a large cohort of animals, male with females, and throughout the trial, we actually see a significant improvement of the memory function of all the treated Alzheimer's diseased animals. So the shorter the time, the better the memory of the mouse is. Showing you how we get how we can improve the memory in animals is just one example of how our technology can change how therapeutics work. As I mentioned, the other goal that we achieve at Synth-Med is changing the outcome for cancer patients. In the case of cancer, there are many ways of treating such devastating disease, but the most recent approaches focus on an immune therapy approach. Here you deliver the antigen of the tumor to the immune system. You trigger the immune response in order to promote the immune cells to kill the tumor cells. This idea is not particularly new, but it has been tried for several years now. The most simplest approach is you take the protein, add an adjuvant to it, and treat the patient with a combination of both the adjuvant and the protein. We have done tests in animals to test competitive technologies and our technology. We do this by taking a mouse again. We transplant the tumor into the mouse and then let the tumor grow. And what you see here on the right-hand side is the size of the tumor over the time. You see, if you have like a very basic approach, the tumor grows relatively quickly. Within 10-15 days, the tumor is so big that the mouse is at the end of its lifetime. Now, let's look at how the state-of-the-art technology works. The LNP mRNA technology. And all of you who went through the pandemic remembers this kind of technology. This was literally the COVID vaccine. Now, it works a bit better, but fair enough, after 20 days, the tumor grows exponentially and the mouse still doesn't survive. With our technology, we can actually expand this suppression of the tumor past the 30 days, and we have even data that go up to 50 days without, by suppressing the tumors successfully in all the animals. You can see how, while it doesn't completely remove the tumor, this technology can actually improve the therapeutic chances for any cancer patients. The two examples, what we are focusing on at Synth-Med, are just examples of how the technology can be applicable in the future. In fact, we have developed a system, as Soline likes to call it, the FedEx of drugs, where we can bring drugs of any kind of format or size to the right target in your body. Our vision for the patient experience is that the patient comes into the clinic, we take about 10 milliliters of blood. That's a blood collection that everybody does regularly for routine checkups. And this allows us to synthesize enough dosages within a day of a process before the blood eventually gets injected back into the patient. Now, I hope I was able to show you a little bit of how I got from a PhD to what we're doing at Synth-Med and got you excited about our technology. But I wanted to end with just saying that I was bleeding for my PhD, but I'm still bleeding to make this red blood cell therapeutics work in the future. Thank you very much.